the eye on the attendee list here and the clock as well. For those of you on spring break, hope you're enjoying some vacation time, well deserved. And thank you for making time to join us if you are away. Today's content should be a treat. All right, I think for the interest of time, well, let's just get going here. So, hi everybody, it's Ethan, Research Director here at Project AP. I'm joined here by Whitney, sort of a program lead at Project AP, and we'll be following up hopefully to get feedback, because we want to improve these roundtables and make the content more interesting, more engaging, and get more participation, and also try to maintain this great ecosystem we have that includes both families and researchers sort of coming to the table in this kind of open science spirit to discuss how we can help all our AP heroes. So, Joe, why don't you just quickly introduce yourself, if that's okay, and Rarebase maybe. I know that some folks on the call will already know about Project AP and Rarebase's collaboration, but if you could maybe just, maybe your slides already do this, but if you can also just kind of recap Rarebase and function and kind of where things, sort of stand, and then take it from there.
Yeah, thanks, Ethan. I'm Joe Bellucci. I'm the Director of High-Throughput Biology at Rarebase. Really excited to talk to you all today about some neuron characterization that we've been doing in collaboration with Project AP at Rarebase. And I'll, as Ethan said, I think go into a bit of detail about what the company does and how our platform operates in the first couple of slides, then try to relate that to the Project AP work. So, as I just said, brief background about the platform to lead off, followed by some details about the cortical glutamatergic neuron imaging and RNA-seq results that we've gathered for primary AP-derived neurons. And then I'll go into some sort of conclusions, how this could potentially tie into the types of things that Rarebase is thinking about from a drug discovery standpoint, and what the next steps might look like from that angle. So Rarebase is a public benefit corporation based out of Palo Alto, California, and we're really focused on small molecule drug discovery for genetically defined diseases. And to do that, we have built out what we call our function platform for small molecule drug discovery. And what the platform does is it essentially tries to integrate biological information across around 4,000 genetic drivers of rare diseases, and then from that information, identify potentially disease-modifying candidate therapies. The platform itself really stands on three main pillars, which are illustrated in the middle of the slide here. The first is cell models. We think this is a really differentiating aspect of what we do. We really try to get disorder relevant and patient specific, in many cases, cell models built and characterized in our lab and in partnered labs. And so what this means is we can take blood from an individual, for example, generate induced pluripotent stem cells from those, and then from those stem cells derive neuronal cultures, for example, and then do a sort of deep characterization of those patient-derived cells. And that's sort of exactly what I'll talk about today, the bulk of the presentation. The second pillar is really drug screening that we're doing in our lab. And this is all done at really high throughput. And I'll talk about this a bit on the next slide, but we're gathering a lot of information around how drugs affect these patient-derived and slightly more broadly disorder-relevant cell models. And then the final pillar, which is equally important, is the computational side of what we do, the analyses that are required to just analyze and also integrate all of the big data sets that are coming from things like transcriptomics of cell models and the drug screens, as you'll see in a moment, is really computationally intensive. And we have a great team set up that's doing great work there. So these three pillars really make up the function platform. And again, the goal is to identify disease-modifying candidate therapies for genetically defined rare diseases. So on the drug screening side, this is really a significant pillar of what we do. And so far, we have screened around 4,000 compounds. These are all small molecule drugs that are either approved or could be clinical stage or might even be very early development. We've screened those in high throughput for their effects on gene expression in a human neuron model. So what we do in the lab is sort of illustrated in this flow chart. We generate scalable neurons, and that's really important, from human-induced pluripotent stem cell cultures. And this is an image of just some of the neurons we've generated in the lab, showing their characteristic neuronal appearance and morphology. We take those cells and put them into what are called 3D 4-well plates. This is where the scalability becomes really important. This is an image or sort of a cartoon of one of these plates. And for people that aren't familiar with these, each little pink dot here is an individual little dish of cells that's about the size of the tip of a pencil lead. So everything's done here in parallel at very large numbers of tests per experiment. And so we've screened, like I said, around 4,000 different compounds in high throughput in this format. And a really differentiating part of our platform from a drug screening standpoint is really that we operate with a very broad readout. So rather than just measuring the effect of each of these different chemical compounds on an individual gene or an individual protein, we really paint with a bit of a broader brush and are looking at overall gene expression in these cells. So we're measuring RNA expression levels using a modified RNA-seq protocol, using next-generation sequencing. And from that, we can then essentially ask, how does a given gene respond to any of the compounds that are in our library that we've tested? So here's a sort of example result where each point on this graph represents how an individual compound is moving, in this case, hypothetical gene X. So on the horizontal axis here, you can see that there are several compounds that lead to increased levels of mRNA from gene X. And just on the vertical, we're showing a significant score. So this is an example set of data that comes out of this drug screening approach for a real gene that's just being masked here. And we can do this across all of the compounds in the library, obviously, but also nearly all of the genes that these neurons express, which makes it a really powerful searchable space that allows us to ask, are there any therapeutically relevant compounds within our library for a particular disorder?
Sorry,
so
sorry, Joe, if
Sure.
I can interrupt. What is the control here, Joe?
So we're testing sort of compound treatment against vehicle for the plot that's generated at right. This is all done in a single cell background. So there's no sort of disorder state versus non-disorder state in this case. These are all neurons that are derived from iPSCs that have been generated from a presumably healthy person.
Okay. Thank you. Thank you, Joe.
Yep. And so there's a couple different ways in which we can point the information that we're generating from these screens. These cartoons kind of illustrate the different gene regulatory strategies that we think can have a therapeutic effect. There are sort of 4 buckets that these fall into. I like to relate this table to the analogy of a car, just for illustrative purposes. So in the first situation, you could have a disorder where there's not enough functional protein. This is something like a haploinsufficiency where a gene, one copy has been deleted. This is sort of the situation where if you don't have enough air in the tires of your car, you can add air to the tires. And the analogy is you're increasing gene expression, making the system perform better. The other side of that coin is that you might have too much functional protein, say from a gene duplication. Same analogy, if your tires are overinflated, you can take air out, make the car perform better. In the case of no functional protein, This would be if, both copies of the gene are inactivated. In the car analogy, this is like if your brakes give out, you can pull the handbrake potentially. It's a different system that results in the same effect of the car stopping in that case. And in the genetics case, this the paralog expression that you're inducing would actually, it's a different protein product that achieves the same functional result in the end in the cell. And then finally, if you have altered protein function, say through a missense variant, you can potentially look at altering the expression of different genes in that protein's pathway to sort of compensate for the deleterious effect. And in this, in the car analogy case, I sort of equate this to if your engine's not working properly, maybe you have an electric motor because the car is a hybrid and you can still make the car go, even though the gas engine is not functioning properly. But I think that's a really important example. Not everybody has a hybrid car. So in many cases, this hypothesis is not really going to hold up. We may not be able to find different pathway nodes or different paralogs that compensate for the genetic cause of a particular disorder. So these are kind of the four strategies that we hope to sort of point our drug screening information along with the disorder relevant information at to achieve a therapeutic end result. So how does this all relate to identifying and potentially developing small molecules against therapeutic targets in 8P? There are, I think, two considerations that we have to keep in mind. One is that chromosomal rearrangements, duplications, and deletions as observed in 8P affect multiple genes clearly because they're changing large regions of the genome. Just to point out, our RNA-seq data has detected pretty robustly around 250 different genes on the P arm of chromosome 8, as you'll see in a moment. So it just gives you kind of an idea of the scale that we're thinking about here. We're not just looking at one specific gene necessarily. It may be a network of genes or a set of genes that might not be related to each other than their presence on chromosome 8P. And the question that derives from that is which gene or set of genes then drives and is responsible for the disorder biology. What we're trying to do here is leverage functional genomics to identify those genes or gene sets that are dysregulated, and from that generate hypotheses around which genes or sets that we could prioritize for therapeutic development. So with that, I'll get into a bit more detail about the RNA-seq results that we've generated on these neurons. First, since I know that there's kind of a diverse audience on the call, I just wanted to review why RNA-seq is important and just sort of generally how it works, just so we can get on the same page about what we're talking about. RNA is really an intermediate between the DNA genome and proteins, where proteins are generally, with many exceptions, performing the actual work in the cell. mRNA sort of sits between the genes in the genome and those proteins, and it's sort of acting as a messenger for creating those proteins. And the key feature and why we're interested in RNA in this case, even though it's not really doing the work in the cell, is that it's a surrogate marker for protein abundance. And from a technical standpoint, it's much easier to measure RNA levels than it is to measure protein levels, given current technology. And RNA-seq allows us, importantly, to compare the expression level of nearly all genes in the cell, and it allows us to compare differential expression between two groups, which will become important, as you'll see in a moment. So the way that the experiment works is that we take those neurons we've generated, isolate the messenger RNA from those cells. For technical reasons and kind of convenience, we then actually convert that back to DNA, which gets sequenced by next generation sequencing. And that next generation sequencing is essentially counting the level of gene expression of all genes or nearly all genes in these cells. So we do some bioinformatics work to align the next gen sequencing results to all those genes. And then we can compare what is the level of all of those genes in an experimental group versus a control group, for example. And in this case, we're really interested in how does the gene expression level across genes compare between the proband and the control cells that we've generated. And does that allow us to identify any therapeutic gene targets? Just before we dig into the actual results from the RNA-seq experiment, I do want to point out, quality is obviously very important to us. This is just a quick slide showing some QC that we've done with immunocytochemistry on these neurons, showing that they are a homogeneous population of cortical glutamatergic neurons. The control cells are indicated at left and the proband at the right. The staining that's been done here is for essentially the neuron bodies and their characteristic dendrites, as shown in green. FOXP2 is a marker of postmitotic cortical neurons, and that's shown in red. And then in blue is the nuclei of these cells. So really what we're looking for is kind of homogeneity of the population. Most of the cells in this case stain for the neuronal marker MAP2. They nearly all stain for FOXP2 at day 7 here. It's around 95% of the cells are FOXP2 positive. And essentially what this shows is that we have a sort of clean population of cells from both the control and proband that we want to take forward into RNA-seq. So having the cells in hand, we then set up an experiment where we're comparing the 8P proband to, in this case, the maternal neuronal sample. Each group has 3 biological replicates. And these, again, are cortical glutamatergic neurons that are, we actually have data from day 7, days in vitro, as well as day 21. And for those of you that are familiar with RNA-seq, the sort of first thing that we do when analyzing the data is run this principal component analysis, or PCA, to basically look at the uniqueness or similarity of any of the samples or replicates in the experiment. And the results of this are shown at right for day 7 and 21. And basically what we're looking for here is that I think the expectation is that the control sample should sort of separate itself and be distinct from the proband, given that these are related individuals and there's chromosomal differences between the two. And that's exactly what we see. The principal components are essentially a way of simplifying the gene expression signature of these cells. And so what this shows is that the control cells, the replicates, are all sort of similar to each other, same for the proband, but that the control and proband samples are very different in this very simplified genetic landscape, if you want to think of it that way, at both days 7 and 21. So this is kind of more quality control, but just showing that the experiment is sort of well set up to investigate any of the differential expression that we're going to talk about in the next couple slides.
So Joe, what do you make of the spread in PC2? I guess could you figure out roughly what PC2 is sort of condensing, what dimensions it's sort of condensing in the data, or is that spread sort of what you've seen before between technical replicates?
Yeah, I think this looks pretty similar to what we see with replicates in many cases. PC1 in this case, I think, represents the vast majority of the variance between these samples. We could potentially dig into that a little bit more, but I think it's pretty standard for what we see across replicates in this case.
Okay, thanks.
And so these are the actual results of, this is kind of, I guess, the key slide, where these are the results of the experiment that have been conducted. And I realize there's a lot going on here. So we're happy to, I think, share all of this analysis as well as the raw sequencing data if those on the line are interested. So just to walk through this, keeping in mind this is proband versus maternal cells, which is important. I'm showing the differential expression of genes at day 7 on the left here and day 21 in the middle. The way to interpret these plots is that the X or horizontal axis shows basically the mapped read depth in the sequencing. So this can be thought of sort of as genes that are expressed more highly are to the right on this scale, and genes that are expressed lowly are to the left generally. And then the vertical axis, or the Y, is showing the test statistic. And this is basically the fold change in gene expression on a log2 scale divided by the standard deviation. So it's not purely the fold change alone, but this metric allows us to separate things that are sort of more significantly dysregulated versus kind of the noise that sits in the bulk of this data. And so this again is proband versus maternal. So anything that is up higher on the vertical axis will be things that are increased in the proband cells, and anything that's lower on the y-axis will be genes that are decreased in their expression in the proband cells. And so what you see is that at both day 7 and 21, there are a variety of genes that are dysregulated between the two samples. I think some of you may notice similarities between the two just by eye from these two plots, but what I've done on the right here is actually compare the test statistics. So essentially taking these vertical axes from both plots, comparing them to each other, and we see really good concordance of the two data sets, indicating that the cells from a gene expression standpoint appear generally very similar, with some exceptions, between day 7 and 21. And there's, I think, two ways to interpret that. One is that the similarities themselves may be interesting. One is that the dissimilarities may be interesting. And I think we don't know that sort of a priori, and it requires a bit more investigation. But another thing to point out here is that there is some kind of critical color coding on all of these plots. Sort of the bulk of genes are represented by gray points. The top 10, just for information purposes, have been highlighted in green. These are the most differentially expressed genes that have the largest test statistic, I should say, not necessarily the most differentially expressed from a fold change standpoint.
Joe, could you maybe just for simplicity, take one gene like relin and maybe just walk through what it means, where it's located, and because we have researchers and kind of family in the crowd, and if we could just maybe take one example and walk everybody through it.
Right. I think it might make sense to do that with sort of this last category, which I think is probably most relevant to this audience, which are the pink points. These are genes that are located on the P arm of chromosome 8. And so, as an example, we could take neurofilament light chain here, NEFL, and there appears to be sort of a statistically significant upregulation of this gene at day 7 in the proband cells versus the control. The converse of that would be CSMD1, which is expressed at a lower level in the proband cells at day 7 and also at day 21. And that shows up on this sort of comparison plot here, showing that, it has diminished expression in the proband cells at both day 7 and 21. There is this general trend in the 8P genes towards increased expression, I would say, this sort of tailing effect where there are these genes that are, there are, I guess, more genes that are upregulated in the proband cell versus control. And I think that's to be expected based on the next slide, which shows the same data, but overlaid on genomic position. So on the left here, I realize this is very small, but this is just showing all chromosomes in the cell and what the differential expression test statistic looks like on the vertical axis for each chromosome. So each point here is an individual gene. The higher up it is, the higher it is expressed in the proband, and the lower it is, the less expression it has in the proband versus the control cells. I think of interest here is chromosome 8, obviously. The P arm on the left here, the shorter arm, you see that CSMD1 gene come back up here as being having a very negative test statistic, as well as I'm color coding here by statistical significance. So it has a high statistical significance of being downregulated. And you see that this sort of gets back to the previous slide where the bulk of the genes on the P arm are expressed at a higher level in the proband versus the control cells. And I believe that this maps pretty accurately to the deletion duplication events that occur in this particular proband sample.
So, Joe, I have a question. Does this child have a deletion or a duplication? Looks like the child has a duplication.
Right. I believe that there is a duplication in this region, as well as a short deletion at the distal end of the chromosome.
Yeah, so this will be an inversion, dupe, and del proband.
Okay.
Also, like globally, you see changes, right, in every single chromosome. And then in addition to that, the genes that are on that AP duplicated don't follow your traditional, that gene dosage, right? They have a diverse range of upregulation, which is super interesting because it's very similar to what we see in Down syndrome. So it's really exciting to see this harmony in between the different aneuploidies, I guess.
Right.
Also, one of the interesting things from your previous data was exist is downregulated in the early stages. And we see that too. And we've never really been able to make any sense out of that, right? Like, because would that mean that, and I don't know if this is a male or female, but would that mean that there's less condensation of that extra X chromosome, or does it play some role? Like it's just, it keeps showing up at this neural progenitor, early neural state. And I don't know if you have any thoughts on that.
That's an interesting feature for sure. I think it also shows up in the day 21 cells as being downregulated in the proband. It's sitting sort of right here. I think it just doesn't make the top 10 in that sample, but it's still, probably top 20 for sure. But yeah, that's an interesting feature. I think there are a variety of genes here that potentially have interesting function in terms of their effect on neurons, for example. And I think one of the follow-up slides to this sort of gets into that a bit more in terms of how we're thinking about that.
Sorry, Joe, mind going back again? I think, could you just, no, the previous slide with the top 10 DGE, could you maybe just, if you didn't, I don't know, maybe you just went through that quickly, but maybe just explain again, what does top 10 DGE mean? And maybe again, if we could look at one of those green genes, like say relin, and just kind of walk through what the implications are. Have you seen that relin come up before, or any of these other, genes that are enriched? Are they unique to AP?
Yeah, it's a good point. So the top 10 genes that are highlighted here are sort of genes that are the most dysregulated between the proband and the control sample that are not on chromosome 8p. So this is, I think, in some ways fairly normal that different individuals, even though they're related, they don't have the same, exact genetic background. There are non-coding genetic variants that can affect gene expression levels. I think you would expect to see differentially expressed genes between sort of any, two people. The sort of flip side of that is that some of these might actually be downstream effects of changes that have occurred on genes that are located on chromosome 8p. Making the connection there is a bit harder from a functional standpoint. We have run things like gene set enrichment analysis to look at pathways that are potentially implicated in this data. And I'm not going to go through that today. It's in the addendum to this deck. We don't see anything super clear there. And I think that association can be difficult unless it's, obviously very clear. But we have looked for that, and I think it's worth investigating more, certainly.
Yeah, one other thing I think I can add to Ethan's point is that, for example, Reelin is something that you'd expect to see in a more mature neuron. So there's going to be genes that are going to be uniquely disrupted because of the state that the cell is into. So there's like so many different variables that you have to account for some of the things that you see.
Right, so
Joe,
Please, anyone jump in who has their
I
hands
had a
up?
quick question for you, Joe. So first of all, you're doing a wonderful job explaining neuronal differentiation and RNA-seq. So I commend you on that. I think you could come teach medical students and graduate students anytime. That's really outstanding.
thanks.
So just a quick thing. So exist is probably really due to the fact that you're comparing maternal to proband. I'm assuming the proband is male, but I actually had a question.
No, proband is female.
Is female. Okay. So if the proband is female, the other reason you might see exist as a differentially expressed gene is because most female pluripotent stem cell lines actually lose exist expression with extended culture. So this is very common. It's actually a big problem in the iPS cell field, and so it can actually lead to X-linked genes being upregulated, depending on how many passages they've gone through without exist expression. So that's something to keep an eye on. If you start seeing a lot of X-linked genes coming up in that proband iPS line, that's something to look out for, because that's probably not real. And then the other thing I was going to say on the next slide, where you had your chromosomes. Most of your chromosomes look pretty good, I think. The one I'm concerned about is 20.
Yeah.
Because I think you have a ISO XQ20. So that's a very, yeah, I think that's what that is probably. I think that's a very common chromosomal abnormality that you can select for in culture, because iPS cell cultures like having an extra copy of the long arm of chromosome 20. And that probably was not in the proband or so if it comes up here, it was only present in the proband line, right?
That's, I think, what it would indicate, yes.
Okay.
I should say that in that GSCA that we've done, the top two, if you look at positional gene sets, sort of the top two most enriched are on chromosome 20. So this sort of duplication looking event here is, I think, real.
Okay. And do we have any other proband lines, iPSL lines from this proband? We had 3, right?
We have multiple clones and we just redid them again, reprogrammed them. So Whitney, right? We might even have more than 3 clones.
Yeah. And
Yeah. So we'll have 3 clones from initially the fibroblasts, and then we'll have multiple clones of same proband from reprogrammed from blood, just in case there's a difference.
Okay. And so, Joe, just to make sure I understand, so the 3 replicates you had were all from the same line. They were not the 3 different proband lines, right?
Correct. Yeah, there are 3 different cultures of the same line.
And do you happen to already have any data from the other 2 lines?
We don't yet, but we would obviously plug that into this same analysis pipeline as soon as it's available.
Also, eventually there'll be a proband versus paternal comparison as well.
That I think is pretty close to being ready for analysis.
I'm just asking so that I know what proband lines are worth submitting for virtual karyotyping. So do you know whether the other 2 lines grow similarly as this line?
Other 2.
It's a lot of shop talk. The other 2
proband lines.
No worry, this is the kind of shop talk we want.
Do they grow similarly or?
I don't know that off the top of my head, but I could check with the lab team about that.
Okay, great. Thank you.
Yeah, I think this is a really good point. 20 is something that we noticed just sort of by eye here. Like I said, it shows up in the gene set enrichment analysis. So I think it's something that we do need to dig into a bit more with these.
Yeah, and if you want to do something really fast, Stem Cell Tech has a kit to look at 12 20 and I think some other chromosome. So you can just do a PCR-based assay and not have to do the whole karyotyping.
Sorry, Joe, did you get a chance to karyotype right after you got iPSC?
These, I believe, were karyotyped. I think this might have been very early days for me on this project. I could check back at that data, but I believe, as standard practice, that's what we do generally.
Right. And it passed. Basically, you did not see any change on chromosome 20 from your karyotype.
I don't believe, right. Otherwise, it would have been flagged and we would have gone with a different clone.
Right. So, yeah. Okay.
Sorry, I thought at least at the provider where these were generated, they were not karyotyped.
They should
Yeah,
have been. I think
Stefan
we were.
is correct, I think. I don't think they've been karyotyped. I'll follow up to be sure, but
Okay, yeah, I'll certainly check on my side too, because we're, obviously doing that as part of routine practice with all the lines that we work with.
It's often better if you have a clone that grows better than the other two, it's often best to put that one aside because it has figured out a way to grow a little bit better. So hopefully the other two are not carrying the same replication.
Right, thanks. That's really helpful. We'll definitely look into that a bit more. I think, just checking the time quickly. So I think that's the bulk of the data that I wanted to kind of dig into, but I do want to talk about the next steps, which would be potentially prioritizing opportunities for small molecule evaluation. That's really what Rarebase is focused on. So a way that we can start to approach this problem is potentially ask, what do the tissue expression profiles of the top differentially expressed genes from that RNA-seq data look like? And can we use those profiles to help us prioritize potential target genes for follow-up? This heat map shows on the vertical axis the top 25 differentially expressed genes, or DEGs, that were from the chromosome 8p arm specifically. You could generate the same plot for the overall differentially expressed genes, but I think we're probably most interested given the genetic background on these. So these were the genes that have the most differential expression in the RNA-seq experiment. On the horizontal axis is, I guess what I'll call integrated tissue expression data from the Human Protein Atlas. This is actually from their single-cell RNA-seq data, but what we've done is averaged it by tissue type. So we take all the cells that are of a given tissue type, average the gene expression level, and then convert that into a color-coded scale here. This is showing the log base 10 of their normalized transcripts per million metric. So this is sort of RNA expression level. So the more green and yellow that you see here, the higher a given gene is expressed in a particular tissue. And I think one of the things that we can do would be to ask, what are the most specific gene targets from these sets of genes that we can generate that are specific to the brain, for example. And I think you can kind of see CSMD1 is actually the highest test statistic differentially expressed gene in this set, and it has very specific, even by eye, brain expression. In the yellow here on expression level in the brain, and kind of purple and blue most other places. We can also do this a bit more systematically by saying or assigning a brain specificity score, I'll call it. This is basically the brain tissue average expression divided by the mean expression in all other tissues. So it's just a way to sort of avoid doing this by eye and kind of get it into a more systematic form. CSMD1, again, ranks very highly using this brain specificity scoring metric. A couple others do pop out here, though. NRG1, which is neuregulin, a gene that's a ERBB3/4 activator with known alternatively spliced transcripts that generate different sorts of growth factors. UNC5D is also a potentially interesting one. This one is a a netrin receptor that's involved in neuronal survival and sort of patterning and growth in the developing brain. So these are, from the literature, very relevant neuron specific targets, and show up highly in our RNA-seq differential expression data as well. So these could potentially be things we would think about, maybe interventions to correct for and establish whether correcting for the differential expression that we're seeing actually results in some kind of disorder relevant phenotype. And the key question is, are any of those targets then druggable, which I think is kind of a broad question. This could be across modalities. I'm not suggesting here that small molecules are necessarily the best way to go about this problem, but there's probably a lot of biology to unpack here, I think, is kind of the key takeaway. One other thing that I should have mentioned is the correction that is indicated on these slides would be, from our standpoint, do we want to increase or decrease the level of that gene to get back to the baseline that the control cells are establishing? So for CSMD1, we saw that it was expressed less in the proband cells, and therefore we'd want to increase its expression level to kind of come back to baseline. So that's kind of how we're thinking about the problem. And just to sort of
Joe, sorry,
Sure.
Can I ask a question? So theoretically, all of these genes are brain-related genes, right? Because they're highly expressed in neurons. That's where you're beginning with. And the whole idea, I'm trying to wrap my head around why brain region specific genes would be more prominent. Is the hypothesis that those brain specific genes becoming upregulated are the problem? Or is there just globally more vulnerability to changes in gene expression in the neural lineage?
Yeah,
right?
I think
Because, yeah.
There's, this is, I think, only one way of looking at the problem, right? You could say, do I want to, if I'm going to modulate genes, do I want to do that specific for a tissue, in this case, the brain, or potentially modulate something that is expressed across tissues and might have different biological effects across those tissues? Like neuregulin, I think, is a good example of that, where those sort of alternative forms have different effects in different tissues. Not saying this is the best way to necessarily approach this problem. This is just kind of an angle one of the angles I think that we're thinking about in terms of prioritizing what targets to go after. In terms of the ranked list here, this is they're ranked based on differential expression. So we're not actually considering the overall expression level in the neurons in this case. I do see your point that these are all genes that are expressed in the neurons, but we're ranking them in this case based on their change between the proband and the control. So they may not be actually highly expressed in neurons in all cases.
Yeah. So within an experiment, let's say a CRISPR-I sort of experiment where you're knocking one thing out at a time, be like informative in this way, right? Like you'd have an idea if like modulating one of these genes on the list would actually be able to, revert the gene expression profile back to what the maternal signature looked like, I guess.
Totally. I think that's a really potentially exciting next step that would come from this is, I'm taking the angle here of small molecule modulation, but the genetic modulations are obviously much more, specific, maybe not as therapeutically translatable, but in the lab, I think that is kind of a key next step is, can we establish the functional consequences, or if there are any functional consequences associated with these differentially expressed genes. And the way to do that, I think, is exactly what you said, is to use something like CRISPR inhibition to knock down a gene, CRISPR activation to increase a gene specifically, and then look at the functional effects of that perturbation.
Cool. Thank
So
you.
I think that's a really exciting follow-up approach that is a really great point. Thanks for bringing that up.
So everyone's welcome to jump in. Definitely raise your hand and we can have a queue or just jump in. Looks like Faye, you're next.
Hi, thanks, Ethan. Thanks, Joe. This is absolutely fantastic talk. I just wanted to say that with regard to sort of Haru's question there, you're looking at brain because these iPS cells have been differentiated into neural cells, right? So if they had been differentiated into gastro cells, so gastroepithelial cells, we'd be looking at a really different profile here. That's correct, yeah?
I think that's what you would expect. That's right.
Okay, thanks.
Yeah, and I think, the brain approach is only one angle that we could take. I guess you could also argue if there is a different sort of tissue specificity that's relevant to, the most important symptom, for example. We could rank order based on the sort of specificity of genes in that particular tissue. So not saying this is the only path forward, just that it's an example.
So I had a question about, I guess, how you would function, how you would query function. Would you want to ask it to find compounds that kind of normalize a whole set of gene expression vectors as opposed to like your driver of interest? Or would you kind of pursue maybe both, like maybe seek something that, like how many molecules would dampen a driver that's seemingly overexpressed versus are there other molecules that might just, even if not a large effect, they might change a large percentage of the expression phenotype. I'm just wondering how to best query function.
Yeah, I think this is the next and I think last slide, other than the conclusions, is sort of like where would Rarebase go from here? And just using CSMD1 as an example, again, not suggesting that CSMD1 is necessarily a key driver. We don't know yet if it has any real biological effect, but if we had an assay, for example, a functional test that showed that neurons were, more or less excitable with this particular gene suppressed, that might be something that we want to follow up on. So if we wanted to follow up on increasing CSMD1's expression, this is what our function-seek query result looks like, where we've got these 12 plots, and what these are showing are what small molecules in our library that we've screened increase the level of CSMD1 relative to a set of genes that it's been established that these are relatively stable across different tissue types in terms of their expression level. So the sort of control gene set, or the housekeeping gene is another way of thinking of it, is shown on the vertical axis here, and the fold change in CSMD1 is shown on the horizontal axis in each case here. And what we see across the panel of 12 is that there are a set of molecules that are in our library that are able to increase the level of CSMD1 while keeping the level of each of these 12 sort of stability genes, I guess is a way of referring to them, relatively constant. So this doesn't suggest that these small molecules are specific for CSMD1 only, but just that we're not seeing these broad gene expression changes across the set of genes that should be relatively stable across tissue types.
Presumably those are like on different chromosomes and sort of like giving you a barometer, of sort of.
Right.
Okay, that makes sense.
Yeah, we wouldn't want to take something forward that, is both increasing CSMD1, but also GAP8, for example. So in this case, these things are able to upregulate CSMD1, at least in our primary screening results. What we'd want to do is take, the set of candidate compounds that sort of follows this horizontal line and is out to the right, and do additional evaluation, mainly in concentration response in wild type neurons first, and then potentially test any lead candidates that we identify in a disorder specific model. But I think a key thing that I kind of have already touched on is generating a functional assay will be really important in the long term. We can obviously test and kind of do confirmatory studies to show that the target gene is coming up or down as we want it to with small molecule treatment. But we don't necessarily know that change in target gene expression will result in a sort of functional consequence for the cell. So we'll need some sort of functional assay to actually show that small molecule treatment can improve the disease state, I think is a way of thinking of it.
And can you also carry to like look for something that would, a molecule that would shift PC1 and so make your proband look more like, well, is that, I mean, obviously you're never going to get it to fully move over, but just nudge it over. Is that another strategy?
Yeah, so that's I think that's where I was about to go. I think that's kind of the next step, is this is only focused on a specific gene. So we would, have to know that CSMD1 in this case is sort of the root cause. We only want to move that gene. Another way of looking at it that we can do, because our data set is so deep, is to ask, can we find a small molecule that sort of best brings the overall gene expression profile back to baseline or more towards a control expression level? That I think is a little bit more of a tall order. It's something we're thinking about, obviously, because we have the data. The odds of finding a smoking gun there, I think, are low, but we might find things that are interesting from that set that merit additional follow-up. So
maybe they move subsets or
Right.
subtraps.
I don't think we'll get them all, but if we get 2 or 3, that could be potentially really interesting to do some follow-up work on. So
Awesome.
A lot of things we're thinking about in terms of the follow-up here.
Perfect. I want to just make sure that we got 6 minutes left. Please, anybody, this is the free-for-all Q&A section. So even if you've spoken before, please ask.
Hey, Joe, you can
hello?
ask.
Go ahead.
No, it's okay. You can go. I've asked questions.
I'm sorry. This is Jean-Pierre speaking. Is it possible to know whether you need to have the same lines tested for 8P cells on the entirety of the glial cells before actually inferring any of the potential conclusions on either the genes or even future functional assays? Because this is a pure, very focused glutamatic, neuron pathway, and 90% of your cells in your brain are basically glial cells of different types. And anybody who's done any sort of neurological, thing, whether it's Alzheimer, ALS, pick your chooses, knows that it's not possible to just focus on neural cells.
Yeah, I think we definitely agree with that. It's a really good point. One of the things that we want to do as follow-up to this is potentially compare this data to the organoid data that was presented a couple of weeks or months ago. I think that would be a really interesting first pass and a potential step towards what you're talking about. But more data is certainly better in all cases, right? So if we had a detailed transcriptomic profile of different cell types, this same sort of analysis could be run. We could look at, are there any conserved gene targets or conserved pathway targets across different cell types, for example?
So yeah, so Jean-Pierre, we don't have glia yet, but we have fibroblasts, we have brain organoids, which certainly do contain glial populations. So I think we're excited to start to cross-reference these data sets. But Hurry, you have another question, please go ahead.
Now, I wanted to ask, for the drug screens that go into the RNA sequencing, how many drugs are you able to do? And just wanted to know the technical, because I think we might want to collaborate with you on some of the other projects that we have, including the AP project.
Right. Yeah, that's great to hear. We ran those 4,000 compounds, 3,800 or so, all in parallel, just in sort of a rolling batch. But we're, doing the same sort of workflow kind of week in, week out at this point. We have a lot of automation that we've deployed at the problem. We're doing everything in 384 well plates. So, we hope to continue to scale that up even more. But I would say that our throughput's pretty good at this point.
And then, so you do the 4,000 drug treatments, and then there's 4,000 RNA sequencing that has happened.
Right. And it's all sort of done in parallel at the same time, just with different indexes that are assigned to each well, for example. So we can batch sequence, many wells and plates all together. And
then, so what's the depth of your sequence, I guess, for each sample?
Yeah, right now we're sequencing to a pretty high depth. That actually is, interestingly, kind of the main cost driver of the entire process, even though NGS costs has come down so much. but we're detecting around 15,000 genes per well per treatment at the moment. We can sort of tune that, based on what we want to see, but that's sort of where we're currently at from a process standpoint.
So you're sequencing like 10 million reads per sample?
Roughly, it's closer to 6 million.
Okay. Awesome. Okay, cool. I'll probably reach out.
Again, no worries. We can go deep. This is meant to be a lab meeting style, so we want to talk about the particulars, but we also want to make sure that we invite people to ask the high level questions or the 64,000 foot as well. Faye, want to come back in? We have 2 minutes here.
I just want to ask, would you consider adding like CSMG1 protein, like recombinant protein, as a control in your experiments when you're looking at your drug screening?
You mean dosing the cells with the recombinant protein?
Yeah, so bringing the level back up.
That could be an interesting experiment to do. We could even do that at low throughput, I think, and it could potentially be interesting if that existed. So
yeah, I don't even know if it's feasible. It's a monster, isn't it?
Yeah. So it would depend on, I think, particular characteristics of the protein too. But I think, as was mentioned before, the sort of genetic perturbation studies could be really valuable as follow-up to this. We do a fair amount of CRISPR-I generally in our lab. I have a lot of prior experience with CRISPR-A as well, and those two tools are just perfectly suited, I think, for follow-up here.
Perfect. Thank you.
Yeah, I mean, it's a great point. We already have those reagents. The Sevens lab is building those CRISPR-I, CRISPR-A reagents, so we can interrogate these drivers in kind of a simpler way than say reintroducing the protein, which surely is one way to do it, but I think not necessarily a platform approach. Great. We're up at time here. I really want to thank Joe for showing up and really taking us on a really fantastic tour of the data and having everybody keep pace. So thanks again, Joe, for representing Rarebase and all the work you've done on behalf of AP Heroes. Thanks everyone for the questions. I think we really are hitting our stride with this roundtable, and it's turning into this this kind of frontline lab meeting style, and we have families and researchers kind of mingling here and sharing ideas. So we want more of this. So don't ever be shy. No question is out of bounds. So thanks everybody. I don't know, Ina, any last words to you? Otherwise, we will make this available through the YouTube channel, and then of course we'll feature this talk in the April research newsletter that will come out on Substack next month.
Thank you, everyone.
Thank
Enjoy
you.
the spring break, everyone. Thanks again for showing up for AP. Bye.